Hi everyone, so in this video we'll talk about polymerase chain reaction. So let's get started. So we'll go through 4 of these pointers in this video, which is the introduction, basic steps of PCR, PCR in details, and then we have the PCR product cloning. So moving on with this. So talking about what is this polymerase chain reaction, or we call as PCR. So, PCR is a widely employed technique in molecular biology to amplify single or few copies of DNA, generating millions of copies of a particular DNA sequence. So, in this, we use a template DNA or any particular piece of DNA which is amplified at certain regions or the particular target regions that we want to target, and we get many smaller pieces or many amplicons of that particular piece of DNA. So this PCR reaction results in the selective amplification of a target region of DNA or RNA. So let's say we take a DNA piece of this, we want to amplify this particular part of the DNA. So PCR is used for that. The PCR reaction will identify that particular target region of the DNA and will amplify that particular part. So PCR has been extensively exploited in cloning, target detection, sequencing. So moving with that, so talking about the basic protocol of PCR and how it is carried out. So components and reagents of PCR include the template DNA, which contains the DNA target which is to be amplified. So template DNA is a very important thing, or the most important thing of PCR. Without the template DNA, we cannot find, or the PCR reaction is unable to find that particular target region of the DNA. So after template DNA, we have the primers. So in this we have 2 primers. One is the forward primer and the other is the reverse primers that is used, which is in the steps of PCR. So primers is used which attaches to the template DNA, or which attaches to the DNA for extension or for polymerase activity. So when, let's say, this is a piece of DNA which is from 5' to 3' and this is for opposite from 3' to 5'. So what primer does is, primer will attach to this particular part of DNA like this, let's say this and this. So now the primer has hold of both strands of DNA, and this will extend this particular piece of DNA to creating more number of amplicons, or it will create the size, or it will extend the size. So primers will keep on attaching and it will help in the polymerase activity. So next we have the tag polymerase, or we can use it as a thermostabler, which is high fidelity DNA polymerase, and it is isolated from Pyrococcus furiosus. Then we have DNTPs, which are the building blocks for the newly synthesized DNA strand. So when primers are attached, as I said, so when primers are attached, these lead to formation of new DNA strands. Alright, so when there is an extension, the extended part are the new DNA strands. So DNTPs are also used, which are helpful in synthesizing DNA strands, so which add to the usefulness of the primer, or which increases the effectiveness, or which increases the enhances the process of polymerase activity along with the primer. So that is why we use DNTPs, that is used for synthesizing new DNA strands. Next we have buffered solutions, which provide a suitable chemical condition for optimum activity and stability of the DNA polymerases. And lastly we have the divalent cations, which can be magnesium or manganese ions. So in most cases we use manganese, and very in less cases you would see manganese ions. So they act as cofactors for Taq polymerase, which increases its polymerase activity, and generally Mg2+ is used, but Mn or manganese ions can be applied to achieve PCR mediated DNA mutagenesis. So this is because higher manganese concentration leads to higher error rate during DNA synthesis. So it is generally preferred to use magnesium ions. Moving with that, so there are 6 main components or reagents which are used for PCR. So with that, so talking about the procedure, how it is carried out. So the most important part that we have come to now is the procedure. So typically PCR is designed of 20 to 40 repeated thermal cycles. So it is carried out generally in a scale of 20 to 40 cycles. So with each cycle it consists 3 discrete temperature steps, which is denaturation, annealing and extension. So as I said, so the PCR overall carries about or consists of about 20 to 40 cycles. So in each cycle, each cycle consists of 3 different steps. All right, so these are the steps which is extended to 5 steps. So there are only 3 main steps that are involved in PCRs, which is the denaturation. So the initial and denaturation you can count as 1. Then we have the annealing, which is 2, and then we have the extension. So full extension and extension is counted as 3. So there are 3 main steps involved in each cycle of PCR, and all the 3 steps are carried out at 3 different temperatures. All right. Also, the thermal cycles are often preceded by a temperature at high range of more than 90 degrees Celsius. So the thermal cycles are often preceded. So these thermal cycles often work at a temperature of more than 90 degrees, and when the PCR is done, or when we get the final product after running it for 20 to 40 cycles, it is stored at 4 degrees Celsius. So it is very important to understand that after the PCR is done, whatever the product we get, we store it at 4 degrees Celsius. So in PCR cycles, the temperature and the duration of each cycle is determined based on various parameters like the type of DNA polymerase used, the melting point of the primers, and the concentration of divalent ions and DNTPs in the reaction. So you can see, so it's not very easy, because the temperature, to determine the temperature and duration of each cycle, we need to take hold of different factors, and thereafter we can guess, or we can understand the different temperatures that might be required, and also the duration for each cycle. So different parameters are the type of DNA polymerase that we are using, which can be Taq DNA polymerase or any other DNA polymerase that we might be incorporating in this process. Also, we need to take hold of the melting point of the primers and the concentration of divalent ions and DNTPs in the reaction. So I will show you a pictorial view of how all of the steps are carried out. So this is the pictorial view you can see here. So it all starts from the denaturation step. So this is the DNA strand that we are using. So for example, let's say this is the DNA strand that we are using. So the first step is the denaturation step, which is carried out at 98 degrees to untangle or to separate the double strand. So DNA is a double strand, as we know. So we need to separate the 2 strands, which would look something like this. So this is the denaturation step, which is carried out at the highest temperature, which is 98 degrees Celsius. Next is the annealing temperature, at which the primer binds to the DNA strand. So as you can see here as well as here, so as the primer binds to the DNA strand, it will help in the polymerase activity, which is the third step, which is extension. So annealing is carried out at a temperature between 48 to 72 degrees. And next we have extensions. So as the primer is bound to the DNA or DNA strand, so it will result in formation of more of DNA strands, as you can see. So this will lead to formation of nascent DNA strands as long as the primer is bound to the DNA. So as you can see, more and more DNA strands are produced, which will result in the elongation of DNA, and more strands will be produced, and thereby the cycle goes on. And the extension is carried out at a temperature of 68 to 72 degrees Celsius. So also there's another picture you can see here. So this is the first cycle, this is the second cycle, the third cycle. So like this, we go up to 30 or maybe 40 cycles. So depending on which part of the DNA we want to amplify and all of that, the number of cycles is determined. So let's say we have will go up to 30 cycles. So the number of copies or number of amplicons that will be produced, or number of nascent DNA strands that will be produced every cycle will increase definitely. So in the first cycle, if we get 4 copies, in the second cycle we will get 8 copies, and similarly in the third cycle we will get 16 copies. So the number will multiply or double up. So in the fourth cycle, same we will get 32, and by the time we reach 30th cycle, or we reach the last cycle, we'll get about 2 billion copies. So you can see how effective this PCR method is, that we can get sampling on so easily and in such a large number, and it would take hardly maybe like around 1.5 or 2 hours to do around the entire process. So that is about it. So I'll show you the last part of this video, which is the basic thermal profile of PCR, which is a very easy one. So you can see there's a steep upgradation of the graph. So you can see a steep slope here, which is the initial denaturation step, which is carried out at the highest temperature in the entire process, which is 98 degrees Celsius. Alright, so you can see this particular graph parallels to 98 degrees. So this is carried out for around, you can see, 1.5 minutes. So this is the profile of each cycle. So how about every cycle goes about? So this is about the temperature graph and the entire process that happened in a particular cycle. So it all starts with DNA naturation at 98 degrees Celsius. Let's say it continues for about 1.5 minutes, and then we move on to DNA primer annealing. So we move to primer annealing, which moves at around at a lower temperature than the denaturation. Let's say around anywhere between 60 to 72 degrees. So here we see the temperature is around 72 degrees. So it carries on for about a minute. So you can see with the graph, you can point that it's about a minute, and then is the last step, which is the extension. So this is the end of the first cycle. So the big line indicates the end of the first cycle, and it all starts, and the same thing happens for the next cycle and the other cycles and the number of cycles that are remaining. So if there are 30 cycles, the same thing will get repeated for 30 times. All right. So the third step, which is the synthesis or the polymerase activity or the extension, which is carried out at around which is at an intermediate temperature between the denaturation and the annealing step. So it's around, let's say, 80 degrees Celsius, and it is also carried for about a minute. So this is a basic thermal profile of PCR. So you might remember this and pause the video and try to draw on a piece of paper, and it will be helpful definitely, and for your information as well. So let's just keep this video till here. Hope you enjoyed this video, and thanks for watching.